It's a great pleasure to introduce my friend and colleague, Dr. Philip Scheltens. And I have a problem. If I list everything Philip has done, he will have no time to be able to his talk. So I'm going to have to cut corners and leave out some stuff. Dr. Scheltens studied at VU University Amsterdam, obtained an MD in 1984, PhD in 1993, and published important work related to brain imaging. He became a professor of cognitive neurology and founded the Alzheimer's Center at Amsterdam University Medical Centers in 2000. And he put together a really remarkable multidisciplinary clinic that provided excellent care, but also became the focus of research. And he directed this until very recently, when he has stepped away in part from the clinic in order to spend some of his time heading a dementia fund at EQT Life Sciences, a venture capital firm trying to help to bring new treatments to Alzheimer's disease. So Dr. Scheltens helped to found the Dutch National Plan Against Dementia. He has helped to lead various European Union projects. He has authored 1,100 peer-reviewed papers and more than 75 book chapters. He is a member of the Royal Dutch Academy of Arts and Sciences. In 2020, he was knighted in the Order of the Netherlands Lion by the King of the Netherlands. He has been elected an honorary member of the European Academy of Neurology and was appointed chair of the World Dementia Council. I've had the privilege to collaborate with Philip as co-editor of the journal Alzheimer's Research and Therapy. So much more that I could say about him, but that will serve as an introduction. And over to Philip, who's going to talk to us about drug development in Alzheimer's disease, some of the problems and lessons learned.
Yeah. Thank you very much, Douglas, and very nice of you to introduce me. And I thought, I mean, this is a topic that sort of has interested me for almost 20 years, I must say, being a clinical trialist as well as the director of the Alzheimer's Center and a neurologist seeing patients. And we have come a long way, I would say, but we are not there yet. Hence also my partial move now to become a director and head of a life sciences investment fund, where I really think that we should make more progress as we have done in oncology. That is much needed also in neurology and especially the neurodegenerative space. And I see Howard also there. Hi, Howard. And it's a real pleasure. And I sort of, this is basically the talk that I gave very recently at ADPD, and it was sort of met with some enthusiasm. And I think it's a topic that a lot of people are really sort of almost troubled with, and engages a lot of discussion. So why have so many trials failed, and why aren't we able to do the right thing, or to make trials a success as we do in other fields? So this is just a very meager attempt from myself and a little bit of the research that we did on this particular topic to present to you. And happy to discuss all the different elements of it, and I hope it will be an engaging 1 hour. This is just my, I will put them on presentation mode. That's probably even better. Yeah, this is my disclosures, and I think I've mentioned all of this. And I'm happy to also lead the Vivorion program, of which Howard is also now a keen leader in the phase 2b study that's running in the US. And we can discuss that as well if you want. So some of the key issues, I think, in Alzheimer's disease clinical trials, Howard, myself, and Jeff Cummings have summarized this in the 5 rights papers. And you can also say the reverse, why so many trials have failed, because they didn't adhere to the 5 rights. So this may all seem very, very sort of as an open door, as we say in the Netherlands. So some of the trials didn't have the right target. I mean, some of the trials didn't even have had drugs that didn't even enter the blood-brain barrier, or had a mechanism of action that was unknown. Many of the issues that you need to tackle in phase 1 or phase 2 were sometimes skipped and then readily going into phase 3, and then with large numbers of people then finding out that the drug doesn't work. But some of the ones that you could actually already have prevented when the drug-like properties, all the MTD and the dose response had been very better established in an earlier phase. I think one of the key elements probably is also the right patient population. So people who really have the disease. So for instance, using biomarkers to select the patient population also as early as possible. I think that's also a lesson that we have learned, especially in Alzheimer's disease, and I will use Alzheimer's as a pars pro toto for the whole neurodegenerative field, because I think it's the most advanced, but also the field where we have learned a lot from. Preclinical prodromal populations are now much more in focus than they were before. And of course, sort of using biomarkers not only to select the patients, but also to look at outcomes and measuring the effect, measuring target engagement. So many of those, you can read them, is sort of captured in the 5 rights of clinical trials. And actually, I've seen now some of the companies using actually this sort of as a cookbook to how to design their clinical trials. But I think there are other issues as well that I've just listed here. Primary endpoints, we can spend hours on it, probably the CDR sum of boxes, the ADAS-Cog, how old can you go, and even the mini-mental, basically scales that were not even designed to measure change over time, but just to measure a specific point in time. So perhaps they are not sensitive enough to measure these changes. And of course, the same goes for some of the functional scales that I won't go into today. One of the other issues that is the variability. Some people call it heterogeneity, and you can call it whatever you want. I've always been very intrigued by the heterogeneity of patients who have Alzheimer's disease. Doing clinic for more than 20 years, you have the experience that patients either sort of progress very rapidly in a couple of years' time, or they don't progress at all. They seem not to progress at all, and they all have the same disease. So there are many factors that come into play, and this may actually also be an issue if you do a clinical trial. You have these fast versus slow progressors, and how do you sort of do you end up with the sort of the same level of progression in one arm or the other arm? And that's a thing that we have studied, and I will present you the study that we did, and you can read it. It was published in Neurology just last year, and we have. Let me do it like this. Yes. Just before I say this, I think one of the key elements that I do think made a difference is the 2018 framework, and we can debate this whether this is the ultimate solution. I'm a very big fan of it. That's not a secret. I think we have made progress in the sense that we diagnose Alzheimer's disease now by its underlying pathology, as we do in oncology as well. And I think the best approach and the best probably estimate of Alzheimer's disease pathology is the presence of amyloid and tau. And since we have these biomarkers now, imaging and fluid biomarkers, to establish these two pathologies in vivo, we can make a diagnosis that actually is not related probably to clinical symptoms or signs or even phenotype, but just identifying the underlying pathology. And I think that's progress. I think it's really progress. And I think nowadays, the clinical trials that we are doing now since 2018, most of them have this biological definition of Alzheimer's disease as its primary basis. So to enroll patients who at least have amyloid and tau in whatever way you measure it. For me personally, I think it's also an important thing to sort of point out that I think now we have come to almost disentangle Alzheimer's disease as a disease with the underlying pathology from the dementia syndrome. And I think many people still equate this, but I think we have now come to realization that Alzheimer's disease can be present when you have completely normal functioning sort of cognition, or you have mild cognitive impairment, anything in between, before you reach the stage of dementia. So I'm personally very much in favor to reserve dementia as a staging denominator and not as a disease indicator. So that's, I think, is progress, and whatever on the right is clinical staging. It's a little bit like the GDS, but 1 point less, and it's very arbitrary, but it does relate to the fact that we just want also to capture, of course, a clinical stage when we enroll patients in clinical trials, for instance. So, and there is much to do about this. You can much sort of variations where you can do it on a clinical scale, you can do it on a clinical syndrome. Doesn't really matter as long as you have the A plus and the T plus as the basis to come to where you go from. So that's one of the things I think will improve and has improved already on the short-term clinical trials. We also know, and this is a very old study actually, there are many of them, that if you have amyloid alone, and if you add the tau to it, or you add neurodegeneration to it, or you add both to it, then people will actually differ in the level of progression that they have. And this is on a mini-mental score, which is completely not sort of adequate for it, but it's just an indication. As you can see, these people all had amyloid, these from the Amsterdam dementia cohort, and the people who have also an abnormal tau level, or also have hippocampal atrophy, or have both, they will actually decline the most fastest. And this is one thing to keep in mind for the rest of the presentation, that this will actually come back as well. And I must credit here Roos Jutten and Betty Times, the first and last author of the paper, and we discussed this issue of heterogeneity in clinical trials, actually inspired by the Emerge and Engage findings, in which the two trials were not balanced in the way that the patients progressed in one or the other arm. So, and this sort of led us to simulate a clinical trial actually in the ADNI population. So this is an example of a trial where you have people within that trial participants who have Alzheimer's disease. And as we know, there are fast decliners and slow decliners, just one of those elements of heterogeneity that we can identify. There are many others, but let's focus a little bit on the decline. In a perfect world, you randomize the patient population to have an even number of decliners and an even number of slow decliners in your placebo and your treatment group. And this will then, if that is the case, then the trial outcome will really sort of reflect a treatment effect. But things can happen, and you can oversample the fast decliners to the treatment group, for instance, and the slow decliners in the placebo group, and then you will underestimate ultimately your treatment effect. Or the reverse, if you oversample the fast decliners in the placebo group, which sometimes happens, so we have seen examples of that in the past, you actually may overestimate your treatment effect, because then it will seem like that the placebo group really declines very fast. Well, in fact, that's not the case, but it has just more fast decliners in that particular group. And we have to realize that randomization can never be perfect and will not always take care of that element. So what we then did, we looked at the ADNI, we selected patients who actually reflected perfectly the patient population that was included in the Emerge and Engage study. So people who had amyloid, based on amyloid PET, for instance, only amyloid, and we had an MCR or mild AD stage with mini-mentals of 24 and higher, and CDR scores of 0.5 and higher, and a follow-up of at least 1 year. And as you know better than I do, then the ADNI cohort has a sort of variety of follow-up. So there was 1 to 9 years in this whole sample. What we then did, we basically, as a statistical experiment, we simulated a clinical trial by just randomly having one part of the group designated as a placebo and one as a treatment, and that doing this 10,000 times or more. And then you calculate the group differences on, for instance, an outcome measure like the CDR sum of boxes at 18 months. We chose 18 months because, again, the eMERGE and ENGAGE and other studies usually nowadays have a treatment duration of 18 months. And then we put that into a group differences plot, as you can see here, with the progression, and we measured the mean and the 95% range. And we did this with many outcome measures, actually, and I will show you. So this is just just an example. This is one of the first individual trajectories and the simulated group differences for all the outcome measures in the total sample. This is the CDR sum of boxes. And you can see that on an average level, on the mean level, people do progress. I mean, that's for sure. Over the follow-up of this time with this particular population with amyloid positivity and mild symptoms on the CDR, you can see that they progress. But you can also appreciate on the spaghetti plot that there are huge inter-individual differences. And if you then plot this into the group differences on, as we did here, you can see that some of the clinical trials, and there are here, the yellow is the ENGAGE, the green is the expedition, etc. The blue is the eMERGE. They actually, some of them fall exactly within that frame. So the treatment difference is actually not different than the natural course of the disease without the treatment. And only one here, the red one here, stands out. And this is the identity on this particular trial on the CDR sum of boxes. And we did the same for the ADRC. You can see here, there is decline over time, although not that impressive, I must say, in 9 years' time. But again, that's probably because there is a huge difference and huge variability again here. And plotted here in this particular graph, you can see again that many of the trials here in the red, yellow, green, magenta, and others are all within this 95% interval. So basically, if the drug had an effect, you couldn't even measure it up and above the noise that was already in there in the natural progression, say, for this experiment. Again, not a real treatment, but just mimicking the treatment. And this is on the Mini-Mental, and you can appreciate here again the same enormous variability here. So then you think, well, as I showed you in the beginning, these people had amyloid positivity. What if we sort of use baseline characteristics, for instance, tau positivity or ApoE4 carriership, or perhaps age? That will probably matter as well. And then perhaps we can reduce the heterogeneity in the variability in this way. And here is an example stratifying on baseline risk factors: tau, normal tau and abnormal tau, carrier of ApoE4 non-carrier. And as I showed you in the beginning, indeed, people who have amyloid positivity and abnormal tau do progress faster. But you can also see that the variability is not that different from the people who have normal tau. Same here with the non-carriers versus the carriers. The carriers do progress faster in this particular sample, but the variability isn't that much less than there is in the non-carriers. And here we'll go through all of the different 3 endpoints again. CDR sum of boxes here, stratifying on age. And to my perspective, I always have sort of thought that the variability in patients who are younger than 65 years of age would actually be less than in the elderly population. But the reverse is actually true. In this particular population, the variability is less prominent in the elderly group than it is in the younger group, as you can see. Again, against my sort of hypothesis, I would again say tau abnormal. Indeed, we saw the progression is steeper, but the variability is also larger. So to the contrary, what you wanted is to select patients on high tau and to reduce the variability, but that's actually not the case. And again, for the carriers versus the non-carriers on this specific measure, the CDR sum of boxes, you can see the non-carriers actually have less variability over time than the carriers have. And well, not to your surprise, probably the ADRC is not that really different. Again, in the younger group, you can see larger variability, even larger than in the CDR, I must say. And in the older group, you have less variability. Male versus female, Again, no sort of real explanation for it, but you see larger variability here in the female population than the male in the ethnic population. Here, higher education, low education, a little bit more variability in the low education group versus the high education group. And again, the same effect for tau, normal versus abnormal. It's not what you want, but it's there what you see. Ultimately, the same for the mini-mental. Again, same story, no difference here. Younger group, larger variability. Abnormal tau, larger variability. Carrier versus non-carrier, larger variability. And here, again, note, I mean, this is the mini-mental. See the differences, the group differences at month 18. So, this is, somebody called it quite a sobering experiment, actually, and that's what it is, I think. So what we found and what we did to simulate more than 10,000 times a large set of patients in a trial-like manner, assuming no treatment effect, no real treatment effect, and we saw already a large variability over time in the CDR, mini-mental, and ADAS-COG, all three outcome measures that are used very often, despite standard and rigid inclusion criteria. At least people were amyloid positive. So they were not completely according to the NIAA. We didn't select on tau positivity as well. But as you can see, as you saw earlier, that probably wouldn't have made a big difference. So if you look at the ZDR, the range of outcome just by the natural progression and the variability is already from 0.35 minus to plus 0.35 in 18 months. So any treatment effect that is a real treatment effect would actually had to be above that measurement, above that specific figure. As I showed you, selecting on ApoE4 positivity or high tau increased progression, but also the variability resulting in an even broader range of effect sizes. So that actually would even sort of yield and needed a larger effect if it had to be a real treatment effect larger than 0.70 on the CDR in order to overcome the natural progression, natural variability. So how to deal with this? This is, of course, the key question. What can we learn from this experiment? And of course, there are many, many ways. This is just an experiment on one specific data set, but I think it's illustrative of the things that happen in real life and in real clinical trial practice. So perhaps we should really be very stringent and say, well, a real treatment effect should really be huge. So let's assume at least a CDR 0.50 or higher is what we think is a true effect. One of my hobby horses, and I won't go into it, is consider perhaps alternative outcomes, more sensitive tests, less variability. In Amsterdam, we designed the CFC, which is a combination of some of the critical elements of the mini-mental, the ADAS, and the Ray auditory verbal learning test, together with the Amsterdam Instrumental Activity of Daily Living scale, put into a combination, and it has shown very nice sort of little variability over time, actually, in some of the samples that we studied. And the ADCOMS is another fine example of a composite score. You may remember the Lilly donanemab trial used the ADRS, which is a combination of the ADAS-COG and the ADL, in which they also showed an effect in sort of a relatively short period of time. I would also say, and this is probably not something that you would be surprised of, that the sample size is extremely important. We have seen this also in subgroup analysis. Smaller samples may actually yield false positive results. I think what we should be thinking of is to have different designs. Perhaps if you look at these spaghetti plots, if there would be a way to compare the individual trajectories instead of group differences, especially when you have smaller samples. And this is also, for instance, done in the DIAN-TU and using a placebo run-in when you have already some knowledge of the trajectory before a treatment is started. That increases power and also may help you find effects that were otherwise could have been blurred. And I think we have seen this also in the DIAN-TU, where large differences in biomarker results were found, but no differences at all in the clinical and the cognitive outcomes. I think biomarker outcomes are extremely important. I think as long as we don't have the right measurements to really measure clinical effects, we are comparing apples and oranges when you look at the biomarker changes versus the clinical changes. And we are sometimes, I think, perhaps a bit too harsh on that. And I think we should realize and be honest that our clinical outcomes are really very not very sensitive. So to discount all the effects of a specific drug based on that insensitive measures is probably not correct. And the biomarker outcomes tell us that there is an effect, there is target engagement, and there is an effect on the pathology, which I think is extremely important. But we can discuss this. This is another way. This is again using all the information from biomarkers in what we call now platform trials, where you have a common placebo arm and you have different, for instance, doses, like in the lecanemab study, and where you use the biomarker outcomes to ultimately define, for instance, your dose and your ultimate readout. And this is especially where sample sizes are small, or you have, for instance, cohorts like the EPAD cohorts, where you have this already, you have your biomarkers that you know of them in the beginning, you can actually do such a platform trial really very well. And this is one of the also the recommendations that was done a couple of years ago by the CTAD task force, actually. And this was again using the DIAN-TU platform as an example of that, to improve the efficiency of the trials, testing multiple drugs and targets in parallel, and optimizing the trial design, again, in view of the limited number of participants. Again, I would say this is especially useful for the trials that we are in, Howard and I, in the phase 2 study, for instance, in the Vivorion, where you have limited a couple of hundred patients only. It's good to be as sort of, original as possible, to use all that you have to look at the effects over time, especially on biomarkers, and adapting then the trial in response to biomarker findings. I think that's what I wanted to share with you, and this is just a brief and just an example of one of the things that we did recently in the center, again, acknowledging the work that my colleagues did. And I will just end here with the pre-COVID last picture we made together. And the group is actually bigger now, but we haven't been able to make such a picture anymore. So thank you, and I will take all the questions that you can think of.